Welcome to the second weekly session of the 2026 Demystifying Medicine series, jointly sponsored by the Foundation for Advanced Education in the Sciences and the National Institutes of Health. This series began in 2003, organized by Dr. Erwin Wynn Arias, who has been the guiding light for this popular series for the past 23 years. I am Harry Malik, Chief of the Genetic Immunotherapy Section and Deputy Chief of the Laboratory of Clinical Immunology and Microbiology at the National Institute of Allergy and Infectious Diseases. I've been asked by Dr. Arias to serve as moderator for today's presentations regarding harnessing the body's own immune system to develop therapies to treat cancers. The Demystifying Medicine series is designed to help bridge the gap between advances in biology and their application to major human diseases by connecting the silos of knowledge and technology that may sometimes separate fundamental basic research and the application of that research to medical advances that benefit the diagnosis and treatment of patients. But equally important, as indicated by the last line in this slide, is that the observations in the clinic also are essential to the advancement of the basic science work needed to discover new principles of biology and new technologies that may be applied to benefit patients. The series is not meant to be simply the presentation of the latest complex science and medicine advances to scientific and medical specialists, but rather the mission is to demystify these topics to make these advances understandable to the non-specialist that may include students, fellows, and interested laypersons, but also engaged specialists in the field. Note the continuing education information on here, and if you want more information about that, you can search Demystifying Medicine CME on any search engine. Next slide, please. This session will examine how the immune system responds to cancers and how that system has been studied, adapted, and re-engineered to develop potent immune treatments for cancers. I will start with the reassuring and encouraging statement that the majority of people do not develop a cancer in their lifetime because our immune system, together with a host of intracellular protective and repair mechanisms, protect us from developing cancers. On the other hand, despite the effectiveness of these systems, 1 in 3 people will develop a cancer in their lifetime because of environmental mutagens and/or inherited factors that may reduce the effectiveness of our natural defenses against developing cancers. Our immune system has evolved strong mechanisms to prevent an immune response to self-antigens. So if cancers expressed only self-antigens and our immune system were perfect at suppressing response to self, we could never develop an immune response to cancer. However, the mechanisms that cause cancer cells to escape growth control often involves biological changes that involve expression of novel antigens or expression of antigens that, while not novel, may be essential to the survival of the cancer. In addition, our immune system is not perfect. This allows natural development of some cancer immunity that may or may not be sufficient on its own to control a cancer, but can be harnessed or imitated by innovative scientific re-engineering toward development of potent immune treatments for cancer that are the topics of today's discussions by today's speakers. Next slide, please. I'd like to introduce our first speaker today, who is Dr. James Yang, a leading figure in the field of cancer immunology and senior investigator in the National Cancer Institute Surgical Branch at NIH. Dr. Yang's academic journey began at the Massachusetts Institute of Technology, where he earned his Bachelor of Science in Biology. He then moved to the West Coast to complete his medical degree at the University of California, San Diego, followed by a full internship and residency in general surgery at the University of California Hospitals. In 1984, Jim joined the NCI Surgical Branch as a medical staff fellow. His commitment to advancing surgical oncology led to his appointment as a senior investigator in 1987, a role he has held for nearly 4 decades. During his tenure, he's been at the forefront of the demystifying medicine theme by bridging the gap between basic immunology and clinical application. His work focuses on the mechanisms of immune tumor rejection, specifically through adaptive cell therapy. This innovative approach involves expanding a patient's own tumor-reactive T cells outside the body to re-engineer them as powerful tools against metastatic cancers. Dr. Yang has been a principal investigator on pivotal clinical trials for renal cell carcinoma and soft tissue sarcoma, and his laboratory continues to develop next-generation reagents to target non-melanoma tumors. In addition to his clinical and research contributions, he's a recognized innovator. He holds multiple U.S. patents for his developments in T-cell therapy and is a long-standing member of prestigious organizations, including the American Association for Cancer Research, the Society of University Surgeons, and the Society for Immunology of Cancer. His work continues to shape the future of how we understand and treat advanced malignancies. In the next slide, I'd like to introduce our second speaker for today, Grigory Altan-Bonnet, who is a senior investigator at the National Cancer Institute and deputy chief of the Laboratory of Integrative Cancer Immunology. Dr. Altan-Bonnet brings a unique interdisciplinary perspective to the field of oncology. He began his academic career in France, earning a Bachelor of Science in Physics from the École Normale Supérieure in Paris and a Master of Science in Physics from the École Normale Supérieure in Lyon. He then moved to New York to complete his PhD in physics at the Rockefeller University. This rigorous background in statistical physics and nonlinear dynamics now serves as the foundation for his work in systems immunology. After completing his doctoral studies, Dr. Altan-Bonnet transitioned into immunology as a research fellow at the NIAID. He later served as a faculty member at Memorial Sloan Kettering Cancer Center, where he held the Bristol Myers Squibb James D. Robinson III Jr. faculty chair. In 2016, he returned to the NIH as an Earl Stadman investigator, itself a very prestigious honor, and he was recently appointed deputy laboratory chief within the NCI's Center for Cancer Research. His research group, the Immunodynamics Group, operates at the intersection of biology, machine learning, and physics. His laboratory seeks to demystify the complexity of the immune system by building biochemically accurate quantitative models. By using advanced tools such as spectral cytometry and the Immunotron, a robotic platform his lab developed, he monitors how millions of immune cells communicate and make decisions. His goal is to translate these computational models into actionable strategies for clinical use, helping to design and optimize next-generation immunotherapies that can be tailored to individual patients. And with that introduction, we can move on to Dr. Yang and his presentation, and I believe he has a patient that he wants to introduce as well. And I'll shut off my part of this, my camera and my microphone.
Let's see, sharing my screen. Are those slides visible yet?
They're coming along. Here we go.
Good now. Thank you.
Okay, excellent. So good afternoon, and I appreciate you all for participating. I'm going to be talking, first, my disclosures. The research you're going to hear about is supported by cooperative research agreements, and I am eligible for patents from the National Cancer Institute. So I'm going to be talking about engineering the immune rejection of cancer. And it's going to start the T lymphocyte, shown here in a peripheral blood smear and in an artist's conception as the main character in this drama. So just to go back and show you how this is developed, if you go back to 1952, the word lymphocyte does not appear in the index of the leading journal of immunology. Nobody knew what the thymus did. Nobody knew what lymphocytes did. They were a black box in 1952. By the 60s, people had started to describe their role in the rejection of skin transplants in mice. And they had identified that there was a class of lymphocytes, the T lymphocyte, that was derived through the thymus. But it wasn't until 1985 to 6 that the first evidence that immune stimulation alone could cause cancer rejection, and that was Steve Rosenberg describing tumor regressions in patients who were given high-dose interleukin-2, which was also known as T-cell growth factor. So the concept I'm going to be talking about, adoptive cell transfer, is disarmingly simple. It's to administer tumor-reactive T cells that are expanded in vitro to cause the immunologic rejection of metastatic cancers. And the theoretical advantages of doing it this way are that you can precisely select immune cells to administer to a patient. They can be rapidly expanded outside the body in an optimized environment. You can use reagents that wouldn't be tolerated in vivo. You can modify or engineer the cells you administer, and you can prepare the cells and the recipient separately to optimize the efficacy of these cells. And so at this point, I'm just going to pause and introduce Dr. Patricia Thompson. Pat was one of our patients. She was practicing OBGYN in South Carolina, but in 2015, she presented with a bowel obstruction that was due to a colon cancer. This was removed, but it proved to involve both her spleen and lymph nodes. And although it was removed, she was at high risk for recurrence. And later that year, metastases to the lung did appear. These lesions initially responded to systemic chemotherapy, but then, as is typical for chemotherapy, they then regrew. At presentation to NCI, she had 8 lung metastases. 3 of these were removed for the production of TIL. These TIL were grown and selected, and after preparative chemotherapy, which I will talk about, she received 73 billion of these TIL, given with 6 doses interleukin-2 in December 2017. So this is the 8th anniversary of her therapy. And so, let's see, I'm going to unshare at this point, and Pat, perhaps you can come on and open your. There you go. Excellent. So thank you for coming. We appreciate your contribution to the entire program. This is a partnership between us and our patients to try and understand this better and make the treatment better. And so I want to ask you a few questions. And one is, how did you first get in contact with us?
So very interestingly, my husband and I are both physicians in a small community in South Carolina. And we had a medical oncologist taking care of us. I had sought referrals and evaluations from MUSC, which was a local university, Duke, MD Anderson, and essentially there everybody was in agreement that I needed to go through traditional chemotherapy in the terms of FOLFIRI. And as the treatment began, it was essentially every 2 weeks. What I underwent was chemotherapy every 2 weeks for about 2 years. Every 2 months, the irinotecan was stopped because I was doing poorly, and they kind of gave me a break. I was always on 5-FU. And in probably August or June of 2017, I was not doing well. So my husband, I kind of had given up and accepted that, probably in this coming year I would not survive. He was very adamant about researching options, and on our own, we looked at the NIH actually on the website and researched the drop-down menus. This is what we have. This is my diagnosis. We got in contact with NIH, and essentially it was a self-referral after failing chemotherapy.
And so when you went through this process with the TILs, harvesting it with surgery and then receiving them back 3 months later, what was that experience like for you?
So I had been so ill and had been on 5-FU for 2 years. I did get a break, and the what was there was a lot of great experiences. A lot of it was positive reinforcement. Hey, we think we've got an option here. The staff, all the people I had interacted with were absolutely amazing and positive. Had the surgery, went home. And, those 3 months of waiting to see, were the cells growing? Was it working? Were very challenging. I was very anxious. Then got a call and said, can you come here in 2 weeks? We think we've got enough cells and we can do the therapy. Interestingly, at that time, I was still practicing full time. And I think my office administrator told me I had about 2,000 patient visits on the books. I also had scheduled through the end of December probably 25 major surgeries. Everyone wants to get their surgery done before the end of the year because they met their deductible. So within 2 weeks, we had to reschedule, redo everything. And I can remember the clinical coordinator saying, you'll be here over Christmas and New Year's. Okay. So that was best news I could get, that I could be there and have a treatment. So the experience, I guess we'll get into this, it was very physically difficult, but everybody was positive, encouraging along the way.
And so what has this been like since the treatment? What's your experience been like with this treatment after you received it?
So, your immune system is quite suppressed. And when I left the hospital, it was very difficult to function. But as time went on, you're healing. And I was not undergoing chemotherapy every 2 weeks where you only have maybe 4 good days at the end of it that you could function well. You just continue to improve for a one-time treatment, which was amazing.
So when was the last time you thought about yourself as a cancer patient?
Well, I mean, it's a scary thing. You still kind of think, when is that other shoe going to drop? So you're always worried about it. And I still kind of think that way, but you're able to start regaining a life and doing a new life. And you think, oh, this is the next chapter. So I think as time goes on, it becomes not so much in the forefront and more in the background.
Okay, so that's very helpful for us. And, one of the things I always told my patients is that the time off of treatment is one of the real blessings of immunotherapy, because when it's working, you don't intervene at all. And so I think that's underappreciated. You mentioned the 4 days out of every 2 weeks where you actually started to feel a little bit better before you had to do it again. That's something we try very hard to avoid, because the longer you're off treatment, I think, is when you garner the best benefits.
Right. I didn't know how bad I felt until after this treatment, I started feeling good again. Because you tell yourself, well, I get to have 4 good days out of 2 weeks. That's good. And then you didn't realize really how bad you were until after this treatment, you're healing and you started feeling good again. Yeah.
Okay, great. So we're now going to talk about how we got to the point that we were able to give you that. And I appreciate your joining us. If you could unshare, I will share the rest of the presentation. But thank you, Pat.
Thank you.
Okay. All right, so the agenda of today is going to be, I'm going to show a very brief history of TIL for metastatic melanoma. Then I'm going to talk about the science behind TIL, and then move on to TIL for the common cancers that most people in this country will die of. So for the non-immunologist, a very brief outline of how a T cell sees its target. This is the T cell here. It's got a 2-chain receptor. This receptor recognizes not the entire protein or target antigen, but a small peptide, a specific peptide excised out of that protein, mounted on an MHC molecule, a major histocompatibility molecule. We all have about 12 of those. And then the T cell sees only this complex. I always think of it as like this is the diamond in a setting. The setting, we have about 12 of them, but every diamond is different. There's innumerable numbers of small peptides that might be mounted on this, but the T cell only sees the complex. And so you need the MHC molecule and the epitope, peptide epitope from the protein antigen in order for the T cell to be signaled. So how do you find tumor-reactive T cells? Well, we knew from laboratory studies that in mouse and in human tumors that these cells are enriched within the tumor they reside in, which is a bit of a paradox, but you could find them within the tumor at frequencies 100-fold higher than you could find them in the blood. And when activated, expanded, and reinfused in mouse models, they could mediate tumor regression. So to illustrate how this works in a human, this is a melanoma that was excised, dispersed into single cells on the left. This is the fresh digest. You see the darkly pigmented melanoma cells, and in between are these small, bright, circular comma-shaped cells. These are the lymphocytes. After a week, you see some fibroblasts trying to grow, the tumors starting to clump up and die, and in between, the lymphocytes gaining ground. And after 2 weeks, it's all lymphocytes. Even this big ball here is a cluster of lymphocytes. No tumor remains, no other cells remain, and this will double every few days in interleukin-2 for many, many weeks. And so this is the way we isolate. We don't really isolate it, we just give it the growth factor, but this is the way we obtain a lymphocyte culture from a tumor. The other thing I want to mention about is lymphodepleting the host. Pat mentioned about being immunosuppressed for a period of time. It turns out the body regulates its lymphocyte population within a relatively limited normal range. The process is called homeostasis. And to infuse and establish a brand new population of lymphocytes, it's necessary to deplete the existing population. And so for all of the T cell therapies I'm going to show you, patients receive 1 course of high-dose chemotherapy with Cytoxan and fludarabine, and that's to prepare the patient for infusion of their cultured lymphocytes. This is just a schema of how it works. A tumor is excised from a patient. We then set up, from fragments of this tumor, multiple individual TIL cultures. We can assay them for melanomas. I'm going to show you that's not necessary, but for other cancers, it is, and to select the best fragments and cultures. And then we expand them rapidly using just interleukin-2 and anti-CD3 stimulation. And then we put them in a bag, wash them, and reinfuse them into the patient who has received this non-myeloablative lymphodepleting preparative chemotherapy. The other issue is T cell fitness. From mouse models, we found that in murine models, tumor-reactive T cells that are multiply stimulated expanded but lost their efficacy in vivo. And this is something that Gregoire will talk about in more detail. This was despite gaining more in vitro effector functions with each stimulation. So there was that paradox. They worked better in vitro, worked worse in vivo. And this was associated with markers indicating increasing differentiation and senescence in these cells. And so differentiated T cells, multiply stimulated, were associated with diminishing proliferative potential and poorer in vivo persistence. And that's the issue of T cell fitness. So when we first started, the first paper was published in 1988, and we were growing melanoma tumors as bulk TIL. So we just did 1 culture, put it in IL-2, the cells grew out, and we gave them to 20 patients with metastatic melanoma. And we treated them with TIL and IL-2, and we gave them varying regimens of lymphodepleting chemotherapy, relatively mild initially. But 11 of the 20 patients had objective tumor responses, and 1 had a sustained complete disappearance of their melanoma at the time of this publication at 13 months after their therapy. And so this was the first clinical report of just an empiric test of melanoma TIL in patients. And it wasn't subtle. This is an X-ray of a patient we treated with melanoma in 2010. You see a big mediastinal lymph node here. You see multiple liver metastases below. And you can see on the right, this X-ray by June 2017, all of her tumor had disappeared and remained gone to this day. And so it was easy to determine that this had activity. This is a patient with dozens, even hundreds of liver metastases on the left when he was treated in 2003. On the right is his X-ray in 2015 with all of those tumors resolved. And it can happen remarkably quickly. This is a patient who had very large melanoma metastases growing under his skin. He was treated, and before he left the hospital, 12 days after his cell transfer, you can see that these have largely regressed already. So in some cases, not most, but some cases, this can also be remarkably rapid. And so this is a summary of the first 93 patients we treated with a slightly varying regimens. And the remarkable thing is this extends out to beyond 8 years, but the same curve actually is now available that goes out over 15 years. And for the first 93 patients, what we saw is some patients didn't benefit at all, shown by them on the left. But as time went on, there was a plateau in which patients survived longer and longer, and it appeared to be about the 30% level. We then decided on a more standardized regimen with high-dose preparative chemotherapy, the TIL, melanoma TIL, and IL-2. And this is a summary of 226 melanoma patients treated. And what you can see here is the dark black line is the summary, and it's slightly better than the last experience I showed you with a more consistent chemotherapy. It's about 40% long-term, and again, this goes out, in this case, to 15 years. But the most remarkable thing is, at the top are the complete responders. If all of your tumor shrinks to the point it's no longer detectable on x-rays, only 2 patients with those complete responses have ever relapsed. And if you look down below, the patients who did not respond, very few of them could be salvaged by any alternative therapy, even though some of these patients did receive what is now very popularly used for immunotherapy, the checkpoint inhibitors. And so this shows that this looked potentially curative because there's this plateau extending from 5 years all the way out to 15 years with almost nobody falling off of this curve. And recently, there was a randomized phase 3 clinical trial in Europe in which they took patients who 85% had had checkpoint inhibitors against PD-1, the most popular treatment for melanoma today, and they were then given TIL as a salvage treatment when they progressed. And what you can see here is the TIL patients, again, are plateauing at 25 to 30% long-term. The patients with an alternative immunotherapy, ipilimumab, none of them are sustaining progression-free survival. And so this was highly significant. And so this trial, plus the work we had done previously, led to this approval of TIL for melanoma recently, February 16th, 2024. The FDA approved the first cellular therapy to treat patients with unresectable metastatic melanoma, which was TIL as we originally described it just 36 years before. So time moves slowly. So the scientific question that came out of this is, what do melanoma TIL recognize? And the early studies found that they would recognize proteins in the melanin synthesis pathway. They would recognize tumor germline proteins, a oncofetal class of proteins that are expressed in development as well as in some tumors. And there was one example of a mutated tumor protein that was recognized. And the question is, which of these are the real antigens? Well, it turns out this is not, because when you effectively attack the proteins in the melanin synthesis pathway, you get vitiligo, loss of pigment, and damage to the eyes and ears where there are pigmented cells. And we didn't see that in the vast majority of melanoma patients who were responding to TIL. Tumor germline proteins proved to be relatively infrequently expressed in melanomas. And so this was unlikely to be the driver mutation for the responses we were seeing. And so that left this category of a mutated tumor protein. And now this was difficult to explore until the advent of tumor DNA sequencing, really in the last 10 years. Widespread use of DNA sequencing on individual patients' tumors led to more studies of these mutated neoantigens, as we call them, the products of tumor-specific DNA mutations. So how often do they recognize mutated epitopes? Well, to investigate this, we looked at tumors and did DNA sequencing. We took all the peptide fragments in those tumors within the correct size range that contained one of the identified mutated amino acids. There are potentially tens of thousands of those, as I'll show you in a minute. And these were listed by the probability using an algorithm that they would bind to one of the patient's MHC molecules. And then we synthesized these peptides and tested for whether they triggered recognition by the patient's TIL. And this is a paper by Paul Robbins that I think is one of the seminal papers in the field. This is a patient who had 570 mutations in their melanoma. That led to 10,000 candidate peptides where a 9 or 10 amino acid epitope, as I showed you, that small piece, contain the mutation. And so for each mutation, there are 19 possibilities times 570. There are up to 10,000 candidate peptides. Now, if you use the algorithm to see which ones are predicted to bind to the patient's MHC molecules, and you list them in rank order from the most likely to bind to the least likely bind, number 10,830, we didn't make them all. We made the first 20. And when you pulsed them onto a target cell with the right MHC and tested them, we got 2 hits in the first 20 candidates. That shows the prevalence of mutated epitopes being recognized by TIL. And so this is, I think, the first molecular demonstration that mutated proteins, consequence of tumor DNA mutations, were generating proteins that were different enough to provoke an immune response. And that explained a lot. This is a plot of 3,000 patients and the mutational burden of their cancers. Each dot is a patient. These are all the different types of cancers. And up here at the top is melanoma. Melanoma has the highest, on average, mutational burden of any cancer due to the UV mutagenesis that's associated with melanoma. And so that explained why melanoma was so good at generating immune responses and immune-reactive T cells. Now, after our finding, this is Paul's paper up here, the next year, multiple papers appeared that showed a strong correlation with the mutational burden of cancers that were being treated with checkpoint inhibitors. Again, a very prevalent form of immunotherapy today, but again showing that the prevalence of mutations was associated with a higher chance of response to these. And this is in a variety of different cancers. But unfortunately, melanoma is not a very frequent cause of death. This is worldwide. You can show the number of deaths from melanoma, the deaths from others. This is about 0.5% of all cancer deaths in the world. And in the United States, it's between 1 and 2%. And so the fact that this worked in melanoma was not addressing the major need. So we then pivoted completely to targeting neoantigens on common epithelial carcinomas. This was the doorway to that because every cancer has mutations. And the question was, would they generate targets for T cells? Unfortunately, they have a lower mutational burden than melanoma, as I showed you. Every tumor would need to be sequenced for mutations in order for us to explore the candidate neoantigens of that tumor. And autologous tumor lines from that patient are typically not available. There was a time in my career where there were only 7 breast cancer lines in the entire world. And so the ability to grow target cells was not available for tumors as they were for melanoma. And so in investigating TIL for the common epithelial cancers, we tried the empirical bulk TIL approach we used in melanoma. 20 patients were treated who had epithelial cancer and there are no responses. But we knew that TIL would differ by location within a tumor. So the next step was actually to dice the tumor into multiple fragments and generate separate independent cultures from different areas within the tumor. But then we also needed a method for displaying the mutated candidate neoantigens on a patient's MHC molecules. And so we could then use them to screen the fragment TIL cultures. And so this is a scheme of how we do it now. Tumor is taken out of a patient, we sequence its DNA, we then encode every mutation we find as either a mini gene where the mutated DNA is encompassed along with a flanking normal protein and DNA sequence. We then string them together into a tandem mini gene just for convenience, or we can synthesize the mutated peptides from these proteins, and then these can be introduced into the patient's own dendritic cell, which expresses all the MHC molecules of that patient, or the dendritic cell can be decorated by incubating it with the synthetic peptides. And now what you've done is you've created an avatar for the patient's own tumor, where their dendritic cells with all their MHC molecules are now displaying all of the mutated candidate targets on their surface on the context of their MHC molecules. We then co-culture them with these fragment cultures I described from the TIL, and we look for either activation markers appearing during the co-culture, such as 4-1BB, or we look for reactivity in the form of an ELISpot. These are wells of an ELISpot assay where each dark purple dot represents a cell secreting interferon gamma in response to the co-culture. So these are the 2 readouts we would use to identify tumor-reactive cells. And this is a piece of this is one plate from an assay that we use to do the screen. At the bottom, there's fragments 1, 2, 3, and 4. There's 20 other fragments I'm not showing you. At the bottom is a TIL fragment culture with very little reactivity. There are some dots over here in this well. These wells represent peptide pools that the dendritic cells are incubated with, or tandem mini genes inserted into the dendritic cells, and then these are the targets of co-culture. And here, tandem mini gene 3 and 4 has a little reactivity in fragment 4. In fragment 3, there's a little reactivity against peptide pools 9, 10, and 11. But fragment 2 has good reactivity against peptide pools 9, 10, and 11, and a little new reactivity against tandem mini gene 9. Fragment 1 is the winner. It has all of these. It has stronger peptide reactivity and both of the tandem mini genes reactivity that we see. And so this is the fragment that we would pick. We would expand it and we would give it to the patient as treatment. The first time we did this was a patient with cholangiocarcinoma in 2014. She only had 25 total mutations, and you can see them listed here, and they were put into tandem mini gene 1, 2, and 3. These were put into her dendritic cell and co-cultured with her fragment TIL cultures. And what you see at the bottom in duplicate wells are mock transduced targets in her dendritic cell, GFP control, negative control protein, but then tandem mini gene 1, when put into her dendritic cell, provoke her TIL dramatically, whereas tandem mini gene 2 and 3 do not. So something within tandem mini gene 1 was responsible, and it turned out to be this mutation in ERB-B2 interacting protein. And it turned out this reactivity, very strong reactivity, was only present in 1 of her 24 TIL fragment cultures. So that's a bit of a tenuous situation. We grew those cells, we gave them to her, and she had a dramatic response. You can see multiple lung metastases and a liver metastasis shown here when we first treated her in October 2013. And this is her x-ray in 2020, and this is her x-ray today. And so she had a complete response. It's always wonderful when your very first patient you ever treat responds, because then you know you're working on something that can work. And you can go to work every day knowing that what you're doing has real potential. And so this was our first patient treated with selected TIL against an epithelial tumor. But we went on to do a variety of others. Initially, we just looked at how often do you find these reactivities. Here's a list of 134 patients. You can see that we found neoantigen reactivity in their TIL somewhere between 67 and 100% of the time. So most of the patients went, overall it was about 3/4. You could find good neoantigen reactivity in their TIL, and the median number of antigens recognized was 2. But on the other hand, of all the mutations screened, only 1 or 2% were foreign enough to provoke an immune response. And so you do have to screen because most of the cultures and most of the target antigens are not players. This is Pat's X-ray. And so she was treated in 2017. As I told you, she had 8 metastases when she came to us. We took out 3 on the right side for TIL, and then these were 3 of her remaining 5 metastases. She was treated in 2017. Her TIL recognized 5 different neoantigens listed at the bottom here, with different phenotypes of cells doing the recognition. And you can see she has small scars remaining, but this X-ray has been stable for over 5 years now. This is the way her X-ray looks today. Actually, last month. This is a patient with breast cancer treated in 2015. And you can see she has multiple large metastases within her liver, shown below. And she had this terrible metastasis in front of her aorta in her chest, growing between her ribs and out onto her chest. And she was treated with a single TIL infusion against 2 mutated proteins listed down below. And she had a complete response. This is her X-ray in 2021, and this is still her X-ray today. And again, this patient has not received 1 drop of treatment since December 2015. So some of the things we learned about this experience is the number of target antigens seems to make a difference. Here is the number of target antigens of patients who had partial responses and non-responses, no response. And the median number here is 3. The median number here is only 2, so not a big difference, but a statistically significant one. And if you plot it somewhat differently, by the number of target antigens in the TIL infused, you can see that the 6 of those patients here had only 1 target antigen in their infusion product, and none of those patients responded. But others had 2 and had some responses, but when you got up to 3 target antigens or more, 40% or more of those patients had objective clinical responses. And so it looks like multi-targeting is an important feature of effective TIL. So the problem with TIL is they can be very labor intensive, take 2 to 4 months to do the screening, and is very costly. And so the other problem is that neoantigen therapy is highly personalized. In those first 134 patients I showed you, only 1 neoantigen, which was a RAS mutation, was recognized in more than a single patient. So every treatment has to be completely tailored to the individual patient. So we asked, are there alternative, more convenient sources of tumor-reactive T cells? And so source number 2 is tumor-reactive T cells that are TCR engineered, T cell receptor engineered from the peripheral blood of the patient. So the patient is getting their own T cells, but T cell receptors found in one patient which target a shared cancer driver mutation can be gene engineered into the T cells of another patient to create tumor reactive cells for that patient. But the patients must share not only the cancer driver mutation, and there are not that many of them that are frequent, but they must also share the MHC type that presents it to T cells. And that makes for T cell receptors that have lesser utility than some of the more generic reagents, but these can be highly effective. The technique of T cell receptor engineering was started in the surgery branch in 2004. This is an X-ray of the first patient to ever receive a T cell receptor found in a different patient, put into his peripheral blood and given to him. And you can see his hepatic metastasis from melanoma is shown here pre-treatment. 1 month later and 10 months later, he's had this partial regression of his hepatic metastasis. And this was first achieved in 2004. But it went on to be more widely utilized and shown to be very effective. This is targeting a tumor germline antigen, NY-ESO-1, with the T cell receptor. And this patient had synovial sarcoma that expressed the antigen and the right MHC type. And she got these cells in August 2010. At the time, she had golf ball sized metastases throughout both lungs. And you can see here in August 2018 that these have largely regressed, as did the volleyball sized primary in her pelvis that shrunk down to a small calcified remnant. So this can be extremely potent using a T cell receptor engineered product. So how do you target common shared oncogenic mutations? Well, we've chosen to target these 2 antigens, and the first one is the RAS family of antigens, most frequently the KRAS gene and its mutations. And it's these 3 mutations of KRAS shown here with substitutions at the 12 or 13 position, which are glycine normally, and substituted with these 3 amino acids. They're present in almost a quarter of all human cancers. And over 85% of pancreatic cancers. And so this is one of the most common activating oncogenic mutations. And so we did develop a library, an expanding library now of T cell receptors targeting these common KRAS mutations, the most common being KRAS G12D, that substitution. And we found T cell receptors that were restricted by HLA-A11. And so when you find a patient that has this mutation and this HLA type, you can make those cells from their blood. This is the patient with pancreatic cancer. She had 2 metastases in her liver shown here. She was treated, and at 6 weeks even, there's dramatic regression, and at 3 months that continues. And so this shows that there is clinical activity against these driver mutation targeting T cell receptors. This is another patient who, just by good luck, had 2 completely different T cell receptors against the same mutation, KRAS G12D. One was a CD4 and one was a CD8. So they're actually presented by 2 different HLA molecules. And so he had pancreatic cancer metastatic to the liver, shown on the left. This is his X-ray as of the middle of June, and this is also his X-ray as of last month, with dramatic regression at 2 years of his pancreatic cancer by receiving 2 T cell receptor engineered cell products simultaneously. The other major target is P53. The TP53 gene is mutated in half of all human cancers, but unfortunately the mutations are disparate. They're multiple. It turns out, though, if you scan across the P53 molecule in cancer, there are hotspots shown here by these spikes in the frequency of a mutation at these locations. And these are the most common mutations in P53 in human cancer. The most common one being here at R175H substitution, which is present in almost 5% of all P53 mutated cancers. And so we generated a T-cell receptor against this most frequent mutation. And fortunately, it was presented by the most common HLA allele in the US population, HLA-A0201. So this T-cell receptor alone would actually be applicable to over 1% of all cancer patients. So this is a woman, a 65-year-old woman with rectal cancer, metastatic to liver, lungs, and bone. She had the mutation in HLA type, and she received this T-cell receptor in her own peripheral blood. And you can she had very large metastases to her liver, shown on the left side pre-treatment. And at 17 months, you've seen this very dramatic but incomplete still regression of all of these tumors from the single treatment given 17 months before. And so again, this is a single T-cell receptor product given to a patient with the T-cell receptor found in a different patient. So looking at the summary of the two side by side, tumor-infiltrating lymphocytes have the advantage of a more diverse repertoire, but it requires surgery to procure them. And probably the biggest drawback is you can't control what's in the product or what is targeted. You can't target the antigens you wish you could, and you can't control how much of the product is the T-cell you want. You can, with TCR engineered PBL, you can select desirable targets, the driver mutations in cancer. You can get a high proportion of reactivity because you make the product. You can even manipulate the function of the product genetically, and that's something that's, I think, the forefront of coming treatments. But it's limited in the number of antigen targets you can use because you have to create each one. So I'm just going to summarize with a review of what I think is happening. What I've shown you is that since the 60s, where there's huge debate as to whether there's any immune response to human cancers, we now know that tumors live with multiple T cells that are capable of recognizing their mutations. But you have to realize that we're losing the battle from the start. When patients come to us, their tumor has figured out how to block every one of these. And so the tumor is no longer under the control of the immune system it grew up with. And so our job is to try and overcome this inadequacy of the immune system and understand the blockade by the tumor. And so TIL, the most obvious and perhaps one of the more important benefits of it is you get a lot more cells. We actually give 20 to 100 times as many cells as are in the entire circulation of a patient when we transfer TIL. We've given up to 1.5 times 10 to the 11th TIL. So you win the numbers game with TIL. But it turns out there are multiple other ways that this tumor can resist immune attack. One of them is it can lose the MHC molecule, and this turns out to be probably one of the more frequent tumor evasion mechanisms. You can lose the MHC that presents the antigen, and if that happens, then all those T cells that required it for recognition are no longer relevant. You can lose the antigen. And some antigens, if they're not essential to the tumor survival, some mutations, you can just lose that protein. And then again, those T cells are no longer relevant to the immune attack on this cancer. And people have also shown that other things happen in the tumor. It can lose the ability to process that epitope fragment and mount it on MHC, or it can lose its ability to respond to interferon gamma, which is essential for some of the responses we see. And so we end up with a situation like this, where the tumor is winning, despite the fact that it's immersed in immune cells capable of recognizing its mutations. But it's winning, but it's defeated them by virtue of the fact that it's growing. And so it has figured out a way to do this. Now, it turns out, though, that not all of these mechanisms are irreversible like the ones I just showed you. Those are all fatal blocks to our therapy. There are reversible mechanisms. And over here, the tumor environment is very hostile to lymphocytes. And so by taking the TIL out of the tumor microenvironment and growing them in vitro, you can overcome this as far as getting good proliferation. There are also inhibitory immune cells within tumors, and those can block. And also the checkpoints, it turns out that the ligands that inhibit T cells are present on the tumor itself. And so the tumor can generate ways to turn off T cells by checkpoint ligands. But we have ways to block the blockers. Growing the TIL in vitro, as I mentioned, the preparative lymphodepletion will deplete the inhibitory immune cells, and there are drugs now that block the checkpoint ligands from binding and inhibiting T cells. And so we give now pembrolizumab, one of these, in association with our T cells, because we found that our T cells do express the PD-1 receptor and are blocked by PD-1 ligand expressed by the tumor in the patients we've given TIL to. And so at this point, we've added that to our T cell therapies. And the last one is the opportunity to add T cell receptor engineered T cells to the mix, perhaps in a new immune attack that the tumor has not seen before. Now, of course, this is still potentially susceptible to inhibitory mechanisms, but it provides us with an opportunity to add a new player to the mix. And so in summary, adoptive T cell transfer can cause the durable rejection of solid cancers. Both tumor-infiltrating lymphocytes and these T cell receptor engineered T cells can cause clinical responses. And tumor-specific mutated proteins turn out to be one of the dominant classes of antigens that are responsible for the immune response of humans against their cancers. But there are still tumor escape mechanisms that represent a major obstacle to success. And a lot of the research we're pursuing now is understanding what they are and trying to develop ways to circumvent them. And I'd just like to acknowledge, of course, all of this is under the pioneering guidance of Steven Rosenberg, who's really been a pioneer in immunotherapy, gene therapy of cancer, and has directed the branch with the major contributions of the other people I've listed. Thank you.
That was just superb. Thank you so much. I'd like to, we're going to hold questions for the end. It's the reason that we have this conference go all the way to 5 o'clock. And at this point, if Dr. Altan Bonnet will present his slides, that would be great. Thank you.
You may be muted, Gregoire.
You may be muted, Greg.
Oh, maybe. What about now?
There we go. Perfect.
Okay, so I was thanking Harry and Win for the invitation to present at Demystifying Medicine. It's a great honor to be here today, and it's a great honor to follow Jim's presentation because a lot of the work we are doing is following up on the shoulders of the giants of the field, and I hope to interest you in what we do. You see my slides?
Yes, coming up now. Perfect.
Okay, so my presentation is going to be about very basic science at some level. We are really trying to build some models of cancer immunotherapy, but our goal is to both contribute to the field and maybe help the surgery branch and others in designing better cancer immunotherapy, but in a more feedback loop, we also want to learn to build models of very complex systems and go back and forth between basic science and more clinical applications. So the field we're discussing is really about systems immunology. So it's something fairly recent, which the idea is that we can really start to measure a lot of things about the way the immune cells interact with the tumors. These days it's very easy to generate fairly large data sets where we can understand which type of cells, what they do, where they are, and so on and so forth. And I'm going to posit that with the recent development in machine learning, in theoretical modeling, we are really poised to maybe understand better the way the leukocytes and the immune system interact with the tumors. And once we understand better, maybe be able to help in engineering an immune response to really get after tumors. So what do we do that for? We want first map the diversity of the response, right? Jim presented a lot of different cases of patients. They are all different. They are all different type of tumors. They are all different MHC, different genetics, different immune environment. At the same time, there are some themes and there's our organizing principle that we are trying to derive to really be able to understand what's happening. So it involves developing some new biomarkers. And I'm going to show you if we are trying to tag along the clinicians and understand how the treatment unfolds, we can really derive some mechanistic insight, really understand how T cells, macrophages, and other immune types can move into the tumors and get activated, and hopefully we can help in optimizing and engineering a better immunotherapy. So at a very fundamental level, a very basic level, we are dealing with a very complex system. When we look at these slides, when we look at the clinical data, you see the tumors, but you see that there are many different types of leukocytes and immune infiltrate which can come in. Some T cells are here to help, some T cells are here to kill, some T cells are here to regulate and try to modulate the immune response. So there are many different ways to address this problem, but at some level, we are dealing with a very complex systems with many things happening at the same time. And our goal is to really build a model of that to be able to understand how it happens. And at some level, we hope that bringing some math to the table, bringing a lot of quantitative approach, we're going to be able to really develop different type of models which help in engineering and understanding the cancer immunotherapy. So at a fundamental level for today's presentation, I'm going to mostly speak about T cells. So it's really following up on Jim's work and Steve Rosenberg's work in the surgery branch. And here I'm showing you a movie which was acquired by Jennifer Lippincott-Schwartz lab and Eric Betzig's labs at Janelia Farm, where they tagged some T cells in red, and they looked at them with super resolution microscopy and look at them interacting with the tumor in blue. And what you see is that there is a very complex dance where the T cell is really like interacting with tumors and trying to decide what to do. And in this particular case, nothing happens. But if we are playing the movie on long time scales, you would expect these T cells to really deliver some cytotoxic granules, perforin and granzyme, and really kill the tumors. So this process looks very awesome and very striking. But what's happening at the molecular level is very difficult to tackle. And as Jim pointed out, is that we really need to understand what type of T cells come in, what type of antigen activations they have, and we hope to build some models to be able to understand this more specifically. So at the fundamental level, what we are dealing with is that most T cells in healthy tissue is going to be interacting with healthy cells, and here I'm presenting myself as a case of self-peptide. This self-peptide are being presented. If you are in this healthy tissue, you don't expect the T cell to do much. Now, if you go into a tissue which has been infected with a virus or with a bacteria, you're going to expect some new antigens to pop up. So if there is something very foreign which is inside this tissue, you expect the T cells to recognize this new peptide as something very foreign and pathogenic, and you are going to elicit a very strong T cell activation. The T cell is going to go and kill these pathogen infected cells, mount an immune response, and you can clear these things. But in the context of tumors, in the context of oncogenic transformation, you're not dealing with self, but rather like something like an altered self. So me being French, I'm going to take an altered self as being Napoleon, and you expect that this altered self, this neoantigen is going to be presented in the context of the self, and suddenly the T cells have a very hard decision to make. Either they're going to recognize this altered self, this mutated self as a strong antigens, and then you're going to elicit a tumor rejection, and this is good. But most of the case, like Jim pointed out in his presentation, all these mutations which are happening on the self genome are being recognized by the T cells in a very weak manner, and it induces tolerance and it lets the tumor grow. So being able to understand when we have a pattern of antigens, most of them derive from the cells, some of them derive from the tumors and from the antigens, being able to understand how the T cells make this decision is something we are still missing. You know, we have some tools we can use to prioritize things, but we are not at the level where we can look at the tumors and really be able to predict whether we're going to get a good immune response. I mean, sure, we need all the screening and all the awesome work that Jim's lab and others are doing. But we hope that moving forward, we're going to be able to push the modeling to be able to really predict this balance between self and non-self activation. So what do we do in the lab, right? If you take some T cells from patients, or if you take some T cells from mouse models and mix them with tumors, you can look at the way they respond. So here I'm showing some data from my postdoc advisor, Ron Germain. And what they are pointing out at the time, like almost 30 years ago, is that when you titrate the amount of foreign peptide, T cells get activated and they start to secrete or to respond and to do things. In this particular case here, they start secreting different cytokines. And we have been developing some model, sorry, I'll try to minimize this thing. So we have been modeling this process, trying to start from the T cell receptors interacting with antigens, going through multiple stages of activation, and all these stages of activations are going to map into different cytokines. So the idea, the conceptual progress we have made in the last 5 years is that if we start to bundle together all these responses that the T cells can do, from producing IL-2 to putting some markers like 4-1BB to expressing different cytokines like interferon gamma and so on, we can take all these readouts as kind of a cue as the way the T cells are wired and the way they can recognize the tumors. So again, each output that the T cells are going to produce is akin to like a neuron, where in a sense there is going to be a dose response. As you ramp up the amount of tumors, you expect this output to be produced. And if you have multiple output, let's say again, IL-2 as a cytokines, or 4-1BB as a marker, or maybe perforin as a measure of a killing, you can start to stack together all these different readouts and try to map the way T cells get activated. What that means is that if we multiplex all these measurements at different time points across many different conditions, we are going to be able to really generate large data sets, which tells us in a way the way T cells are wired. And what it looks like in the lab is that we have been developing at the NCI a platform called Immunotron. So it's a robotic platform in a whole pipeline where we can do the assay, which is typically done in the surgery branch in other labs, where we can collect T cells and tumors or APC, mix them together, put them in our robot, and then the robot is going to go and take measurements by itself, in particular at different time points, and give us this kind of very complex map and complex dynamics of the way T cells are activated. So here at the bottom left, I'm showing you like a summary figures where we're looking at the T cell on the right and the tumor on the left, dancing with each other. The T cells get activated. They start to differentiate into and move into different states. And at the same time, they're going to influence the way the tumors are going to grow and respond. And we hope to be able to really capture the dynamics of this process to be able to build a model of T cell activation. So this is what it looks like in the lab. If you come to the lab in the building 37, you can come and visit and see this robot running. Hopefully it runs constantly. So we collaborate a lot with the clinical lab in building 10. We collaborate with other labs over VNH, and we can really generate this assay and let the robot run and automatically generate data sets to be able to really map all the different conditions. And there are a lot of tools being developed from the machine learning field these days. On the right, this is a graphic interface that Suraj Achar was a grad student in the lab generated. It enables us to really multiplex and kind of build a very large scale observatory of what T cells can do. And that way to really generate large data sets that we can use to build some models. I enjoin you to come to the lab if you want to see the robot in action, and it's kind of the future of what we do in terms of experiments. So very quickly, we end up with this kind of data sets. Here I'm showing you a training set. So we took some T cells from a mouse models. We activated them with different quality of antigens, which we kind of model the different type of tumors that these T cells may encounter, different level of MHC, different time point, and on the x-axis, different readout, which correspond to different state of activation or different cytokine being produced by these cells. And you see that if we just take this data and just represent them, project them into a low-dimensional space, there is no rhyme or reason. It's very complex. There's a lot of things happening. 4-1BB might go up, interferon beta gamma might go down. It's very complex, and there is no way to really tackle this data in a smart way. We can make a lot of observation, which can be very useful moving towards the clinic, but we hope to do better and to really be able to capture this awesome complexity, but at the same time build really a model of what the T cells are doing. So there I'm going to make a digression to give you a very quick introduction about machine learning. So what I'm stating is that at this stage, when you look at the field of immunology, we are very good at making a lot of measurements. So I'm really comparing that to where the state of astronomy was before Copernicus, which is that we could measure all the angles. So if you go to the Greek astronomers or the Arab astronomers, they knew very well the position of Saturn and Jupiter. We had this very complex geometry to understand things, but you see that it's not a very useful model. It's not a very useful model because the Earth is in the center of the universe and you don't really have the right set of variables. And we really hope to push the field of immunology towards a more cogent model. So again, Copernicus revolutions put the sun in the middle of the universe, and suddenly you get very simple trajectories. You get a model which is way more useful if you are trying to fly a spaceship or if you're trying to understand what's going to happen next in the system. Well, I would posit that at this stage in immunology, we're a little bit in the left field where we can measure tons of different things. We don't really know how to organize the data. And we are hoping to use machine learning to get into that stage where suddenly we're going to have a very mechanistic model on understanding of what T cells are doing. And that way we're going to be better at kind of piloting the immune system towards fighting tumors. So I'm going to make a small digression, a very simple primers for people who are not familiar with machine learning and AI or artificial intelligence. So this is a very general introduction just to give you a sense. And I gave that actually in France. So for that reason, I made an analogy with French Revolution, and it's also to state how artificial intelligence can be revolutionary. So imagine that I want to build a model of the French Revolution, and I get all the pictures of all these players, and I really want to understand who is doing what, right? So you have many different participants. Some of them are British, we are fighting Napoleon. Some of them come from the Dominican Republic or Haiti and did another revolution in the colonies. But the point is that we would like to build a model of the complexity of this era. And there are many things that you want to address. You want to understand who come from the army, who was a civilian, man or woman, is this person more on the side of the republic, or is more on the side of monarchy, and so on and so forth. Well, if we take all these images, we can start to break them down into small bits. And for example, we can build a model to try to understand about the hat that some of its revolutionary were building. So you break down the data into lots of small pieces, and you ask, is there something which looks like a hat? Yes, no. You combine them together, and then you make a decision, and you say, yes, this person seems to be wearing a hat. He must be a soldier, right? You can do that with many different questions. So you feed different aspects of your data. So again, here, I'm just showing you images, but this could be like a T cell phenotype. This could be the state of the tumors and so forth. So you make a lot of observation, and you're going to decide whether this is going to be up and down on these neurons, and you feed them into a decision. And the key aspect of machine learning is that some tools have been developed in the last 10 to 15 years, which enables you to train the system and to learn all these weights in such a way that you're going to be able to project this very complex pattern, this very complex set of data into a simple decision. And in that way, you're going to be able to learn a model. So at the end, you can train a model to learn different things, military versus civilian, monarchies versus revolutionary. And at the end, you're going to have a powerful code which can take data. So in this particular case, again, images of French revolutionaries, but more interestingly for us, phenotypes of T cells, status of the tumors, and so on and so forth. And again, based on these codes, we can really start to break down the data into small bits and try to learn a model and try to learn something very important for the immune system. So back to the immunology of cancers, we're trying to build a model of T cell response against tumors. And again, what we have is a very complex pattern of antigens being presented by the tumors, different MHCs, different type of antigens, different co-receptors. All these things get read by the T cells, and then it activates an immune response which protects cytokines. And this geometry is very much like what I just showed you with the images. We can really start to take the output, for example, the cytokines here, or some markers, and try to break it down into different type of response to really build a model of the way T cells get activated, and hopefully to be able to learn the way T cells respond to tumors. So that's what we did here. We took the cytokine being produced and measured in our robot. We feed it into a neuronal net. We squeeze the data into a very low dimensional space to make decisions according to the quality of antigens, and that way we can really learn a model of T cell activation. So what it looks like on the left is that with the raw data is a very high dimensional space. I cannot show you 27 dimension, but just like 3 cytokines like IL-2, interferon gamma, that TNF that Jim mentioned is good readout of T cell activation. Suddenly, when you measure all of them at once at different time points with different antigens, you get this very complex pattern, but maybe regular. And when you take this, feed it into a neuronal net and try to squeeze it into a low dimensional space, suddenly it's going to look into something extremely manageable, something which looks very regular. So suddenly, a good antigen, a hot tumor is going to give you this kind of blue activation, and the cell tissue is going to give you the red activation. So we can take this very complex space and really tackle it into low dimensional space. And indeed, then we can parameterize it. So again, high dimensional space of response with cytokines and markers and so on, squeeze into a low dimensional space, which is called a latent space. And then we can parameterize it with a single parameter. And then we can go back and check that this simple parameter that we call an immune velocity, so it's kind of the temperature of the way T cells get activated, completely correlates to the way we would have measured it in a traditional way. And we can really train the model, which in one scoop is going to tell you how T cells get activated. But not only that, this model was trained with mouse T cells, so something very artificial and designed to be simple. But this model, which has been trained from mouse data, can be applied to a clinical sample. So these are NY-ESO specific T cells, the one that Jim was presenting in the context of clinical trial. And exactly the same model is going to be able to be applied. In that way, we can really rank the different type of antigens being presented on these tumors and being able to predict that depending on the type of antigens, you're going to get a very strong response or a much weaker response simply by having used all this data from the mouse. So I think we are making very good progress in being able to tackle the complexity of immune response. We know that many things are happening at once. T cells getting activated, recruiting macrophages, trying to kill the tumor is going to fight back, and so on and so forth. So if we can measure all these parameters and project it by using neuronal net and artificial intelligence, suddenly we're going to get a very objective parameters of the way the immune response happens. At least we can rank and predict what's going to happen for patients. And hopefully we can learn how to really move the needle on these things to be able to commute a weak immune response into something which is way more powerful. So moving along, we took these models and we applied it to CAR T cells. So CAR T cell is one hot topic as well in the field of cancer immunotherapy, where people are trying to engineer T cells with the chimeric receptors. So the part which is extracellular is an antibody which can recognize the tumors, and then it's linked to signaling domain borrowed from the T cell. So it's a bit of a chimera which contains some aspect of T cell and some aspect of antibodies. And again, we took our models learning this latent space and this low dimensional space, and now we can put the CAR T cells on top of that. And what we find is that CAR T cells are pretty good at killing tumors, but they look very weak in terms of T cell activation. They look like one of the weakest antigen possible. But not only that, the CAR signal, when it's mixed with the TCR signal that every T cell brings along, can be antagonized, and we can really have some negative interaction. So I'm not going to go too much into the details. This is an awesome collaboration with Naomi Taylor and Nir Alisha in the pediatric oncology branch. But again, using our modeling, we can go back and analyze some of the CAR product and really understand why sometimes this CAR product fail because they get antagonized by the natural response of the T cells. So this was kind of a bummer in terms of CAR T cells. At the same time, it opens a new possibility in something that we're working on actively with Jim and Naomi following along. This is the idea that we can really engineer CAR T cells in a better way. So instead of taking a simple T cells which contain the receptors, we're going to make a dual T cell which contains both a receptor which can recognize the tumors and also a CAR. But we're going to pick up a TCR, we're going to pick up a T cell in such a way that can recognize the tumors in a positive way with a strong antigens, and it's going to recognize healthy cells with a very weak antigens, and this weak antigen can really antagonize the CAR. So what we ended up with is something which is the best of both worlds. I'm going to skip this part for the sake of time, which is that we can now, based on our modeling effort and based on all this math that we have been able to do, we can go back to the clinic and really engineer CAR T cells which have the best of both worlds. So we're going to have a CAR T cell with both a CAR and a TCR, and we pick up a TCR, we can recognize the tumor as a neoantigens. In that context, the TCR is going to accelerate the CAR T cell activation and really boost the way we can kill the tumors. And at the same time, we're going to pick a TCR which recognizes the self very weakly. And again, Jim very kindly provided us with some of these examples where we had a TCR which recognized P53 as a neoantigens and recognize the self P53 as a self-antigens, as a weak antigens. In that context, the TCR is going to start to antagonize the CAR. It's going to act as a brake, and it's going to start to protect the healthy tissue which presents the normal P53 in a regime which is really protected. So at the end of the day, we hope to correct some of the shortcomings of the CAR. Typical CAR, for example, against CD19 can go and kill leukemia cells, and they tend to kill the normal B cells. Not a big deal because we can live with B cells. But when you take this CAR and you try to, for example, target HER2, we know that HER2 is being presented indeed on breast tumors, on different carcinoma to very high level, but there is always a low level on the lungs. And indeed, you have a lot of cytotoxicity because these CAR T cells, if you don't do anything very specific, are not going to be specific enough for the tumors, and they're going to really push the patient to respiratory failures. So we hope that all the modeling I showed you and all this fine-tuning of the way the TCR can really recognize the tumors and discriminate from the self is going to enable us to really fine-tune the way this CAR T can be designed with, again, a boost of activation in the context of the tumors and a break and some antagonism in the context of healthy tissues. And that's mostly what I wanted to present today. Again, I hope I caught your attention to point out that we know how complex tumor microenvironment can be. There is an adaptive immune response which happens, which we want to trigger. There's an innate immune response which happens at the same time. And being able to engineer all these players and being able to really fine-tune all these different cytokines, all these different pathways, the way T cells get activated, the way they can recognize the tumor, the way they can communicate with macrophages and so on, opens up a lot of possibilities that people are exploring in many different ways. In our case, we are really trying to use systems immunology and more math modeling and machine learning to really be able to tackle this complex microenvironment and really, hopefully, get back to the clinic and collaborate with Jim and his team to be able to really design different TCRs and better immunotherapy in the long run. So again, very complex math, but we think that given the fact that we can really measure a lot of things these days, and there's a lot of tools developed from machine learning and AI, we're going to be able to make good progress to develop some very simple model, and hopefully we can really communicate with clinicians and get the ball moving on that front. So this is the lab at present time, with many collaborations, specifically with Naomi Taylor's in POB again to develop this dual TCR CAR. Lots of help from Jim Yang and Steve Rosenberg to have access to TILs and to be able to test different TCRs. And all the machine learning is done in collaboration with a group from Paul François from the University of Montreal. And then and of course, a lot of help from new tools that the CCR and the NCR allows us to develop based on robotics. And to conclude, again, we have a lot to do in terms of future directions. We need to be able to activate the immune response in a smart way. We need to be able to circumvent all the obstacles associated with the tumor rejection. So again, on the left side, I presented my side of the story in terms of being able to model T cells, being able to understand how TCR activated and seeing how it maps into different type of activation of T cells, different stage of differentiation, and being able to build a model of this T cell activation. And Jim?
Yeah, so we've been looking at the tumor side of the equation, but it's really amazing how in the last 50 years we've gone from major debates about is there any immune response in human patients to their cancers to understanding this incredibly complex dance that occurs. It's more like a half fencing match, half dance between the tumor and the T cells over periods of 5 or 10 years as the interaction evolves. And the patients we see are the ones where the tumor has won. The tumor is growing, the immune system is not doing its job, and our job is to try and unravel that and reinforce the T cell and understand how the tumor cell escaped and try and defeat that. And so it's remarkably complex, as Gregor has just shown you. but it's a step forward in our understanding of what's happening in this tumor microenvironment between these two principal players.
Thank you, Jim.
That was just superb. I'm very grateful to both of you, and I think what was quite clear is that there's synergy between what you both are doing in the clinic and in this dance of lymphocytes and trying to understand a model system. One of the questions that perhaps we can start with, and I've sort of learned this from Wynn, is to ask both of you, even though you work together and you interact together, and perhaps I'll start with Jim. What would you hope that Gregory would be able to provide you that would help you to further optimize and perhaps move forward with what you're doing faster?
Yeah,
in the clinic.
While he was talking, the idea flashed across my head that his tools would be perfect for a fundamental problem that I've had for a decade, which is, there's two types of T cells, CD8 T cells and CD4 T cells. CD8 T cells generally are cytotoxic, and their efficacy generally correlates with their ability to kill the tumor in the dish. But CD4 cells, they make cytokines. They don't interact with the tumor. We've proven that in mouse models. They interact with normal antigen-presenting cells to do something that makes the tumor go away. And we've proven clinically that CD4 cells can be sufficient to treat human cancers. We have 2 protocols that demonstrate that. And so I do not understand in the faintest how a CD4 cell treats a cancer. And so
I noticed that you had, in some of what you presented, you combined both CD8 and CD4 kill cells that and I actually had wondered, why you were doing that, why that helped.
Well, because clinical experience has shown us that a CD4 cell alone is sufficient to treat a human cancer. We had a protocol where we had a class 2 restricted receptor that we put into peripheral blood, treated 17 patients, only 11 at the maximum dose, and 4 of them responded. And so that's a pure CD4 class 2 restricted activity that we created and gave a patient that caused tumor regression. And we just recently had another patient who's responding to one against the P53 epitope. So we know a CD4 cell is sufficient to treat a human cancer, which is an important thing to understand. There's very little evidence that proves that. But what it does after it sees an antigen-presenting cell and gets triggered, I have no idea. I have no idea how it gets rid of the tumor, because mouse experiments show if you knock all the MHC out of the tumor, knock it out with CRISPR, you can still treat it with a CD4 cell. You cannot with a CD8 cell. And so I don't understand that. And Gregoire, tracking what's happening between tumor and T cells and APCs in the environment would be enormously informative, because I don't know what cells are participating. I don't know which ones are being turned on, what functions they're performing, but something happens and the tumor goes away.
Right. So first, I'm going to jump on that, because first, Rémi Bosselu has a fantastic study where they dissect the role of CD4 in this MHC-independent way. So stay tuned, or reach out to him, because he has a very fantastic story about that with some imaging with Ron Germain. It's a very nice story. And then to answer Harry's question, it's exactly what the synergy is, that we want to be inspired by Jim works, and again we have to be very humble about what we can do, but like learning from the clinician what seems to work and trying to put a bit of math maybe to explain at the mechanistic level gives us a chance, one, to understand better, and maybe manipulate things, and maybe titrate things, find the right stoichiometry between CD8 CD4s in a way that maybe would be very difficult to explore in a systematic manner without the math. So I would say math is mostly just another microscope. It's just a way to extend the way we understand the system, and hopefully find a regime of parameters which models what Jim sees in the clinic, maybe explains it, and at best, really, we can reproduce it, and it becomes a target we want to hit, right? And,
Yeah,
and again, I want to be very clear, but we are really standing on Jim's shoulders and Steve Rosenberg's shoulders. This is all these examples of cure are extremely inspiring, and we hope to really learn about immunology, and hopefully, it fits back and helps in the treatment.
Yeah, the clinical experience says when it works, says this is important. This can work. This is worth pursuing. But you can't do a clinical trial for every T cell. It's impossible to prove that this T cell works and that doesn't by doing a randomized clinical trial. So you need the tools that Grégoire talked about. But the clinical experience is valuable to say, this is worth studying with those tools.
Yeah, I noticed that an aspect of what Grégoire was showing, it looked like he was able to qualify potent antigens versus less potent antigens, and then the sort of interactions and pulling apart why that is the case. And I was wondering if at some point, knowing which patients had tremendous responses and which patients failed, whether his tools, even if they don't tell you why, they might provide predictive things that you can start ahead of time, so that this pill or this pill cell receptor is going to work in this patient because of these markers that perhaps you've identified.
That's right. So as you point out, like historically, we have some markers of T cell activation which are fairly good, but they are not precise enough to be able to rank all these antigens with this fine grain. Right. And having this like very high dimensional space, it's not just more markers. It start to deconvolve different APCs, different quantity of antigen, different quality of antigens, and we really start to rank the antigens in a more objective manner. We have not feedback yet onto Jim's project, but we really hope that, again, having trained this model, we can go back to some of the library of TCR is looking at, or other people at surgery branch, and be able to have a more objective measure of what they look like in a more absolute way.
So I'm going to build on a question from the audience to Jim, which is that you've now got FDA approval, which means this is potentially commercially available. I'm assuming that there's companies or a company that's beginning to develop this, and is this, how expensive is this? Is this going to be doable? And could the things that Grégoire is doing allow you to work out ways of reducing the cost? Because it's both time consuming and likely expensive. How expensive is this likely to be?
Yes, the company that has brought this to use is Iovance, and it's for primarily melanoma, although they're starting to look at other histologies. Yeah. And so melanoma is clearly the leading contender because it's a plug-and-play TIL product. Yep. You don't have to do the screening. There's so many mutations. It's like having 3 to 5 times as many shots on goal. Well, right. More will get through eventually than with the other tumors. But the expense of it is very high. I think they estimate that trying to give a patient this treatment will cost at least $500,000. But the average advanced cancer patient will spend almost $1 million a year as they die from their metastatic cancer. And so if you think of that, it's a one-time treatment. If it works with a high enough frequency, you obviate all those other costs of supportive care, hospice, ongoing salvage therapies, first, second, third line chemotherapies. And so I think the economic analysis of a one-time treatment that can cure you, you have to also factor in, if you're cured, as Pat will tell you, you'll continue paying your taxes for another 20 years. And so there are economic advantages that I think outweigh the cost if it works frequently enough.
Yeah, I mean, we in the immune deficiency gene therapy arena are facing the same problem where, as well as the sickle cell approved gene therapies, which is There's lots of complaints about high cost, but if you think of lifetime costs versus one-time cures, it balances out. But there's still issues. There's
Yeah, I've always
problems
said that this is the worst business model in the world. If you want to make money in pharmacy, you invent a pill that costs 1 cent to make, and a patient has to take for the rest of their life, and you sell it for $50 a pill. The worst model is to invent a treatment that costs $100,000 to make that you use once. So I can understand the obstacles to commercial exploitation of this. Quite frankly, that's not the highest thing on my radar screen.
So I'm going to build on another question that's come in from the audience, a little bit twisting in another way. I know you didn't talk, Jim, at all about CAR T cells, but that's the yin and yang of the two T cell approaches, with the CAR T cells not being natural T cell receptors at all, but rather is a sort of a jerry-rigged system that takes something from antibodies and something from T cells, and as Gregory pointed out, he did bring up T cells at one part in his lecture, and they clearly work, but there are issues. They run out of steam quicker. They have a lot of other issues that need to be addressed, and I was interested in what you've begun doing with your CAR T cell clinical colleagues to try to fix some of these things, but I can translate that into a more general question. Why do you think CAR T cells have been so effective in sort of blood cancers, or actually in very specific blood cancers, but haven't, people are trying hard, and there's been some successes, but they really haven't done the job the way your TIL cells have done with solid tumors. Do you think we're at a crux point where we're beginning to discover how to make those CAR T cells do some of the magic of the TIL cells?
I think Gregoire's and Naomi's approach, trying to spare normal tissues, will potentially expand the utility of CARs, but there's a fundamental problem. CAR cells are manufactured in a pharma factory somewhere, and it takes many years to do it. And so they have to be against a constant target. They have to be against a target that does not change between patients. And that almost obligates them to target a normal self-protein or cell, like a B cell, malignant or benign. And you wipe out all your B cells, you can survive. But every other solid tissue in your body, it's hard to knock it out and survive. Your bowel, your lung, you can't knock those out. And so if you're targeting a normal self-protein, which is almost obligatory for a CAR, you're going to have trouble. The T cell, our factory is the thymus. We make a new T cell. We make 10 to the 15th T cells, and we get to pick the ones we're going to use. We do not have to use an inviolate target. We have a factory that can make, 10 to the 12th T cells. And so that is our factory. And so we can then target things like mutations, things like a mutation that may only occur in one patient in a million. And so that's why it can apply to solid tissues, because you can now attack things that are on the solid tumor tissue, but not on the solid normal tissue. And so this is a fundamental difference between these two modalities. And that I think it's going to be very hard to bridge it. Now, I hope that the combined T cell break and acceleration model that Gregoire showed will spare normal tissues, but it has to be very potent. You can't afford a little leakage against your lungs or your heart or your brain by a CAR that's targeting a brain tumor or a lung tumor or a heart tumor. It's got to be really tight. And so that's what I think is yet to be demonstrated and will determine whether this works or not.
So one other question from the group. As you know, sometimes people have simply used allogeneic transplant to try to harness donor versus tumor effect. Although the questioner asked the question, how does TCR engineering differ from graft versus host? I think I'd like to twist that a little bit and really ask the question, the anti-tumor effect that you get from donor versus tumor, how do you think that differs from uh, uh, from 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 TIL cells, or for example, TIL cells that you've created from somebody else's T cell receptor, but engineered into a patient's cells? Do you think it's a similar mechanism?
No, I think it's the same fundamental difference there in that the graft versus tumor effect is mostly effective in hematopoietic malignancies, and we think it targets minor histocompatibility antigens, but those are not mutated. They're just differentially expressed on certain hematopoietic cells and other normal tissues. So you're targeting something that could be on any tissue potentially, but has a differential expression pattern. It's always trying to just trying to hit that little window of safety between killing something you need and killing something you don't want. And so I think that's the problem, and that's why it's not a very strong reactivity. On the other hand, I can devastate R175H mutated cancer because there's not another normal cell in the body that expresses that P53 mutation. And so I can really take the gloves off and use anything to attack that. And so I think that's the reason it works in solid tumors better, but it's also the reason that the graft versus tumor reactivity won't, because you got to keep the gloves on. If you take them off, you're eventually going to hurt something you want.
So again, I'm bouncing around a bit, but I'm trying to build off what both of you have done. So you talked about checkpoint inhibitors, and now you're beginning to use them with your TIL cells. And I know that some people even using their CAR cells are starting to use checkpoint inhibitors. My question is, do you think that checkpoint inhibitors without cell therapies are really sort of doing what you're doing at a different level? That is, they're unmasking the potential of the patient's own TIL cells.
Yes, they talk about taking the brakes off of the anti-tumor response with checkpoint inhibitors, but if you're driving a Yugo and not a Ferrari, you can take all the brakes off you want. It's just not going to make your tumor go away. Right. And so the key is they're working with the endogenous T cell repertoire. With adoptive transfer, you can change that. You can transform that repertoire in a way, but it may still need the brakes taken off of it. And so it turns out that when they work by themselves, it's one of those lucky patients whose tumor inhibitory mechanisms I showed, that the dominant one for one of those T cells was simply PD-1. And if you block that alone, the shot gets through. But if it's lost MHC or other things, that's not going to fix it. And so you just have to be lucky enough to have that inhibitory mechanism, the dominant one, for your anti-PD-1 to work by itself. But if you change your repertoire, you can alter that equation.
So I would say, like, from the math side, the dominant mode is what Jim describes, which is mostly the TCR and being able to get the Ferrari and very strong TCR. The PD-1, in a way, is a rescaling of this axis. So again, if you are lucky, you were already at the edge and you're going to go above the threshold and you get going. But you are doing way better if you can really design a good antigen and a good TCR. It's way more potent. So it's a matter of scale in a way. It works, obviously, PD-1. It works well if you have a lot of mutation, but you are going to do so much better if you can really have a good antigen and a good TCR.
So getting into the cost and time issue, it would be great if what has been described could be given to anybody who possibly has a tumor that might respond to this. But at least in the near term, that's not likely. And so if there may be patients who are going to respond to checkpoint inhibitors, Gregory, do you think that your system might be able to predict which patients are going to respond?
So we hope so. I mean, we're working on that. I would point out that engineering the cells is very important, but even the cancer vaccines, right? If we can even bypass, making the cell outside of the body, if we can really identify good, bespoke antigens for each tumors, for each patient, and there are evidence in the context of pancreatic, for example, where it has worked mostly for stage 2 pancreatic, but still they were able to sequence the tumor of the patients, identify some potential neoantigens, and they didn't even try to engineer the T cell outside. They just injected this peptide and were able to elicit a good immune response. So being able to do that very quantitatively and being able to predict would reduce the cost, reduce the time scales, but we're not there yet. I mean, we
The real revolution will come when pathology departments buy in, because
Right.
we throw 1 million tumors away in this country every year into formalin. It's a complete waste of information, and it would help those patients potentially. And so not only should those tumors be subjected to some very basic but informative examinations, but we should MHC type and molecularly type the mutations of patients' cancers automatically when they come out of that patient's body. They shouldn't have to send away to some company and spend extra money, sometimes without insurance, to get some genetic information about their tumor. So the pathology departments have to buy into the idea of genetically typing tumors quickly and broadly, MHC typing patients, and subjecting those tumors to a little immunological interrogation to determine which ones are going to benefit the most from immunotherapy.
So most pathology departments aren't set up to do that. Are there some commercial entities already out there that if they wanted to do this, could do this?
They're starting to be. There are a couple of the companies that genetic testing on tumors, what you send to them, are starting to add MHC typing if you send them a tube of blood as well. But you have to order that and pay for that separately. But the biggest tragedy is the 1 million tumors dumped into formalin every year.
Yeah.
I would point out that there is progress on the machine learning side where people have been able to take these formalin sections and train a model. So maybe not to identify mutations at the level Jim would need, but at least to start to characterize the immune environment, right? Being able to look at the morphology of the cells inside these sections. And with enough data, you can really start to quantify how many T cells, how many NK cells, what type of leukocytes you have, even though these measurements were not done for that. So we might rescue these 1 million sections, Jim, but not to the level you need, but at least get a bit of information about the type of immune settings you have in these tumors.
So,
Yeah,
so
Bill,
go ahead, please.
Bill Gasby is a pathologist working in my lab, and by looking at just basic RNA-seq data on the tumors we take out, he has a profile that can greatly enrich for tumors that will grow effective and reactive TIL and therapeutically beneficial TIL. So that's a very simple, look that any lab could do that, but it's not something that pathology departments are doing yet. I'm sure it will come, but it's very slow.
So sort of building on that, as a very theoretical question, I'd like you to speculate on. Do you think if you look long and hard enough, that every cancer has a likely potential target? I realize very speculative. I'm just pressing you on this.
I'm a theoretician, so I can say whatever I want, right? I would say
good.
yes.
Okay.
So I would say the immune system can recognize
that's
everything.
a strong argument for saying everybody should have a bit of their tumor put away, cryopreserved or something like that.
I'll tell you, I would.
I would go even beyond that. I would say every tissue in our body has a distinct repertoire of T cell and distinct signatures. And this has been adjusted over the years as you reach homeostasis. But understanding that means that maybe we would be able to trigger whatever we want in whatever tissue we want. We're not there yet at all, of course, but if we had this level of depth in terms of TCR repertoire and understanding of the homeostasis of the immune system interact with every single tissue, tumor would be just one more tissue in a sense. But that's theoretical.
Yeah, that's very helpful. That was sort of the answer I'd hoped to hear, which says
Jim is smiling. I hope you notice Jim is smiling.
It's both very encouraging and discouraging because it really seems like somehow we need to have people storing their tissues so that they may benefit from these things. So one other thing, you've made some great advances now in cloning T cell receptors that then can be used more broadly, so that you've got a subset of patients where you can take their lymphocytes and turn them into the TIL cells you want, but it's obviously MHC restricted. And it sounds like you've mostly been working, and as you know, I chair the Institutional Biosafety Committee, so I review all of your things that are, and I've sort of asked this question in the past, but I'm going to ask it now. How are we going to, most of your things have focused on HLA O2, and that, anywhere between 20 and 50% of people may have that, which means they may benefit, but it still leaves a lot of people out. And I was wondering how you might envision, broadening this out to more HLA types.
Well, eventually, I think there'll be big libraries where you can just type in your MHC, type in your list of mutated antigens, and they will tell you what T cells are available for that. We already have probably several dozen, but we'll need thousands to cover,
Yeah.
And so, but the bigger question is, what's a good T cell receptor? We found redundant T cell receptors that are not equivalent in a variety of models. And so Gregoire will actually be helpful in identifying the best T cell receptors, because that's equally important. If you have 10 of them, which one do you use? You can't run a clinical trial for each one. And the clinical trialing of these has to change. The approval of what you can do in a patient with T cells and T cell receptors, I think has to change, because you can't clinical trial every one of them. It's hard to even do a phase 1 trial if you're restricted by HLA and mutation. And so you have to have a different standard with what is reasonable to try in a patient with metastatic cancer, and you need multiple receptors. I always say the example for me is trying to do a phase 1 study on a 3-legged stool and then make you do one leg at a time, before you get to try all 3.
These are regulatory issues, yes.
Yeah, that's not going to work.
And many of us in the gene therapy field, which is what this is a form of, are the whole concept of umbrella and trials and program, and the idea of platform approaches. Hopefully, the FDA is not going to ask you to do everything on every single T cell receptor.
It becomes impossible. It becomes
impossible, right?
Yeah. Personalized medicine has led to that. It's so personalized, you can't do large trials anymore like we used to do with a single reagent.
No, and the regulatory process is going to have to approve the process rather than the individual receptor and the individual antigen, I think.
I would posit that if we formalize all the assays we can run in advance, maybe we'll get to a stage where we know what to look for. I mean, again, I'm not a clinician, so I cannot extrapolate, but issues of toxicity and so on, maybe we could capture that in model system, which can be run on a small scale, and then that would speed things up quite a bit.
Well, this has been just profoundly interesting and very, very encouraging. I mean, I think the future is. Wynn has some questions. Yes.
Yeah, I'd like to get back to what the patient said and get your opinion, both you, Gregoire, and Jim, about the fact that, I mean, here you have a physician whose husband's a physician, and they have some sort of a thought process that they could look for the NIH as a possible source of treatment of their disease. But she makes the point that they had to do that on themselves. It didn't, I mean, if I heard her correctly, it didn't come from either her general physician or even the cancer centers that she went to. So it seems to me that this is a huge hole in this whole enterprise. If it's dependent only upon some people by luck of the draw becoming candidates in a clinical trial, which is not widespread throughout the country in multiple institutions. What are your thoughts about how that obstacle of making at least what's done at the NIH accessible or available or knowledgeable? You don't even read much in the general literature about this. what we heard today, for example, not so much the high technical details, but the accomplishments, the goals, the possibilities. I think the general public is probably totally unaware of it. So what are your thoughts about how to deal with that, which granted is not strictly a laboratory question?
No, I think it's a real problem because I think if I was a colon cancer patient, I would be overwhelmed with the protocol choices and have no way to evaluate them. You know, you can say that, well, look for early stage positive results. Well, those early stage trials have to also accrue patients. So somebody has to get into this trial before anyone knows anything about it. And that's really almost impossible, you know. So it's just that there's a great need and so many patients who are looking that any of these trials accrue anybody. But it is a completely random process. And I don't really know how to fix that because we want many, many options out there being trialed, but we also want to know which one's the best one. And I don't see a resolution between those two conflicting needs. It's a problem I don't have an answer to.
It doesn't, your answer I appreciate, but it seems to me it doesn't address the question of what could, in your view, what would you like to see the NIH do to make what you're doing at least more aware to the cancer public as well as to the general public?
Yeah, I think that every major university I know working in this field has a publicity machine that works 24/7 for both the investigators and the trials that broadcast what they're doing and look to get in contact with patients. The NIH, has the communication department, the NCI has a communication department, but that has actually been pared down to almost nothing. And so we're not good at that. We've never been good at that. But it costs money. It takes effort, and we don't charge money. So it becomes a bit of a difficulty staffing and pursuing that. But that's how other institutions have managed this.
Well, I think these talks today were spectacular, and they certainly opened my eyes to some of the exciting work that's going on literally right across the courtyard. So I'm sure all those attending are very gratefully appreciative of your comments, and certainly, Harry, for your directing all this and your cogent questions. Those were my comments. I turn it back to you, Harry.
Well, I'll close by saying, absolutely, I was blown away by both of these presentations. And I know a lot in the area, but I learned a lot of new things today. I really appreciate it. Thank you.
Thank you for
again.
inviting us.
Absolutely. Thank you.
Thank you all.
Okay.
Bye-bye.
Bye-bye.
Thank you all.